Hello, welcome to this presentation. I am S.O PienS. I want to give a presentation on nerve muscle physiology. This presentation will help you in two ways. Number one is it will give you the quick overview of what nerve muscle physiology is all about. Number two, it will help you to have a quick revision, of nerve muscle physiology. So now let's have the outline of the presentation. Number one, we have introduction. Number two, we have physiology of nerve. Number three, we have the physiology of the muscle. Number four, we talk about the types of muscle. Number five, we talk about muscle contractions. Number six, we talk about membrane potentials. Number seven, we talk about synaptic transmissions. Number eight, we talk about pathophysiologies. Right? Now let's start. Introduction. Nerve muscle physiology is otherwise known as the physiology of excitable tissues that deals with the study of the functionality of excitable tissues, which are majorly the nerve and the muscle, and how impulses flow through these excitable tissues to drive them to work. That's what nerve muscle physiology is all about. Now, a nerve is a collection or aggregation of nerve fibers outside the CNS, that is, in the PNS. A nerve fiber are the axons that extend from the neuron cell body. Two major nerves leave the CNS. They are the cranial nerves and the spinal nerves. The cranial nerves are 12 pairs, the spinal nerves are 31 pairs. These nerves are distributed all over the several regions of the body. They contain afferent, which is sensory, and efferent, which is motor nerve fibers. The human CNS, that is the central nervous system, contains about 100 billion neurons plus the glial cells in 10 to 50 times the population of the neurons. So you can imagine how populous the glial cells will be, which is like 10 to 50 times the population of the neurons. The neurons are the basic building blocks of the nervous system, and their specialized functions are the integration and transmission of nerve impulses. Now to muscle. A muscle is a collection of muscle fibers. A muscle is excitable because it responds to stimulus by conducting action potential, which drive it to contract. So as a nerve is a collection of nerve fibers, so also a muscle is a collection of muscle fibers. So both are actually tissues. Now a muscle can contract because of the presence of the contractile proteins, which are the actin and myosin. The third excitable structure is a glandular cell, which responds to stimulation by releasing secretions. Now let's go to the second part, which is the physiology of nerve. A neuron is a structural and functional unit of nervous system. A neuron has cellular processes that we call axon and dendrites. Axon is more referred to as the nerve fiber because thousands of it can actually organize as a nerve. So axons are more organized than dendrites. That's why axons are more referred to as nerve fibers. But in actual sense, the axon and dendrites are both nerve fibers. Now, a neuron cannot undergo cell division simply because of the absence of centrosome. Now, centrosome is an important organelle that contains two centrioles important in cell division. That's for the formation of mitotic spindle. So absence of centrosome consequently causes a neuron to be incapable of undergoing cell division. A neuron with several poles or cellular processes is called multipolar neuron. An example is a motor neuron. A neuron with two poles or processes is called bipolar neuron. An example is granule cells and ganglion cell in the retina. Then a neuron with one pole that later divided into two is called a pseudo-unipolar neuron. Now let's talk about the neuron proper. A neuron has two parts. We have the cell body and the nerve fibers. The cell body is the soma or pericardium, okay, that integrates impulses. The nerve fibers, they transmit impulses. The dendrites and the axon. Dendrites bring in impulse towards the cell body. Axon, takes the impulse away from the cell body. Now, axon can be myelinated or not. It can have small or big diameter. It can release specific neurotransmitters. Okay, nerve conduction in myelinated axon is called saltatory conduction, and in non-myelinated axon is called continuous conduction. Okay, impulses flowing in the axon towards the axon terminal, that is towards the synapse in the right direction, is called autodromic conduction, while impulses moving in the opposite direction, that is towards the cell body, is undergoing what we call antidromic conduction. Okay, now this conduction, that is the antidromic conduction, cannot cross the synapse to re-excite the presynaptic neuron. That is the principle of one-way conduction. So a neuron cannot re-excite its presynaptic neuron. Neurons communicate at the junction that we call synapse. Okay, that will be discussed later. Now, glial cells. The glial cells are the Schwann cells that myelinate nerve fiber in the PNS, oligodendrocytes that myelinate nerve fibers in the CNS. So Schwann cells in the PNS, oligodendrocyte in the CNS. Okay. The third topic now is the physiology of muscle. Over 600 muscles are present in our body. Skeletal muscle is the most abundant. Okay. Now, muscle is made up of muscle fibers or muscle cells that we call myocytes, okay, which are cylindrical in shape. Muscle fibers are surrounded individually by a connective tissue that we call endomysium. Muscle fasciculus, that is small collection of muscle fibers, is surrounded by perimysium, okay, and the whole muscle mass or tissue is surrounded by epimysium. These arrangements are well delineated in skeletal muscle. Okay, a muscle fiber has numerous small fibers that we call myofibrils, which are present in the sarcoplasm of the muscle fiber or cell. Other organelles in the sarcoplasm, that is in the cytoplasm of a muscle cell, is what we call sarcoplasm. The nucleus, the mitochondria, the sarcoplasmic reticulum, the ribosomes, the Golgi complex, the glycogen granules. Okay. Each myofibril is made up of myofilaments, which are the thick and the thin myofilaments. The thick myofilament is majorly myosin. The thin myofilaments are the F-actin, the tropomyosin, and troponin system. Please note that all these features are majorly in the skeletal and cardiac muscle. Since myofibril is a long cylindrical structure, myofibril is actually a cylindrical structure that is located inside a muscle fiber. It is segmentalized into sarcomere. A sarcomere is a segment in a myofibril, okay, and a sarcomere is a structural and functional unit of the skeletal muscle that contain the contractile elements, the actin and myosin, in appropriate arrangement. Okay, a sarcomere extends between two zed or disc lines, of myofibrils. Now, based on the arrangement of the contractile elements, each sarcomere features two major bands. We have the A band, which is dark, so dark. The second letter there is, as A, so A for dark, and I is the light band, so light. The second letter there is I, so light band, I for light band. Okay. Now, I band has the Z line, A band has H zone, which contains M line in its middle. Okay. The sarcotubular system is a system of membranous structures that invaginate into the sarcoplasm of the muscle fiber. Okay, it includes the T, that is the transverse tubule for rapid transmission of impulses, and that is the longitudinal or sarcoplasmic reticulum tubules for the storage and release of the calcium ions for muscle contraction. Okay, now let's talk about the fourth topic now, which is the types of muscle. Three major types of muscles. We have the skeletal muscle, simply because it's connected to skeletal tissues like bones and cartilage. That's why we call it skeletal muscle. Cardiac muscle, called because it's found in the heart. Then smooth muscle, called because of its appearance. It's smooth, it's not striated. and is found in visceral organs. Now, smooth muscles are of two types. Okay, we have the multi-unit smooth muscle and single-unit smooth muscle, or visceral smooth muscle. The multi-units are closely related to skeletal muscle. They are found in the intrinsic muscles of the eyes, that is the ciliary muscles and the iris muscles. They are also found in the blood vessels. They are found in the erector pili muscle, then the detrusor muscle of the urinary bladder. Okay, that's where you see the multi-unit smooth muscles. The single unit or unitary or visceral smooth muscles, they're related to cardiac muscle, that is in having gap junctions. They are majorly located in the GIT, in the uterus, in the ureters, and the wall of the respiratory tract. Alright, now let's talk about the differences between these types of muscles. Now we're talking about the features, the skeletal muscle, the cardiac muscle, and the smooth muscle. Feature 1. The shape of the muscle fiber or cell. In skeletal muscle is cylindrical and unbranched. In cardiac muscle is bifurcated. Okay. In smooth muscle is spindle shaped and unbranched. Okay. Now the second feature is action. In skeletal muscle is voluntary. The action is voluntary. In cardiac muscle is involuntary. In smooth muscle is involuntary. Okay. Now the control of contraction, that's another feature. Control of contraction in skeletal muscle is neurogenic. Control of contraction in cardiac muscle is myogenic. Control of contraction in smooth muscle is neurogenic and myogenic, so it's both. Okay. Now let's talk about the nerve supply. For skeletal muscle we have somatic nerves. For cardiac muscle we have autonomic nerves. For smooth muscle we have autonomic nerves, that is the para and the sympathetic nerve fibers. Okay, those are the visceral nervous system. Striation. Skeletal muscle is striated. Cardiac muscle is striated. Smooth muscle is not striated. Presence of syncytium. No syncytium in skeletal muscle. There is syncytium in cardiac muscle. There is also syncytium in some smooth muscles. Okay. Now neuromuscular junction. Neuromuscular junction in skeletal muscle is well defined. In cardiac muscle is not well defined. In smooth muscle is not well defined. Then the speed and duration of contraction. The speed and duration of contraction is fast and short period in skeletal muscle. In cardiac muscle is intermediate. In smooth muscle is slow but long period contraction. The sources of calcium ions. In skeletal muscle we have the sarcoplasmic reticulum, right? In cardiac muscle we have the sarcoplasmic reticulum plus extracellular calcium ions. Okay, so that's important for contraction. Then smooth muscle is majorly extracellular calcium ions. Calcium binding protein. In skeletal muscle we have troponin C. In cardiac muscle we have troponin C. In smooth muscle we have calmodulin. Calcium regulatory protein we have calsequestrin. Okay, in skeletal muscle. In cardiac muscle we also have calsequestrin. Then smooth muscle we have cardesmon. Okay, those are calcium regulatory proteins. The nature of sarcolemma, that is the plasma membrane. In skeletal muscle it lacks many hormone receptors. In cardiac muscle it contains a variety of receptors, that is the alpha and beta adrenergic receptors. In smooth muscle it contains a variety of receptors. The same thing for like in cardiac muscle too. Gap junctions. Now it's absent in skeletal muscle, it's present in cardiac muscle, and it's present in some smooth muscles. Depolarization occurrence. In skeletal muscle is by stimulation. In cardiac muscle is spontaneous, that is due to the presence of pacemaker cells. That's the SA node. Then in smooth muscle is also spontaneous due to the pacemaker cells too. That's the interstitial cells of cardiac. Okay. Fatigue. Fatigue is possible in skeletal muscle. It's not possible in cardiac muscle. It's not possible in smooth muscle. Summation. It's possible in skeletal muscle. It's not possible in cardiac muscle. It's possible in smooth muscle. Okay, please note that. Tetanus. Skeletal muscle can be tetanized. Cardiac muscle cannot be tetanized. Smooth muscle can be tetanized. Okay. Nature of T-tubules. T-tubule in skeletal muscle is long and thin. In cardiac muscle is short and broad. In smooth muscle is rudimentary T-tubules. Sarcotubular system. In skeletal muscle is well defined, well developed. In cardiac muscle is well developed. In smooth muscle is rudimentary. All because the first two muscles, that is skeletal and cardiac, they are striated. Myofibrils and sarcomeres. is present in skeletal muscle, likewise cardiac muscle, but absent in smooth muscle. Troponin system is present in skeletal muscle, present in cardiac muscle, absent in smooth muscles. Resting membrane potential is stable in skeletal muscle, stable in cardiac muscle, but unstable in smooth muscles. Okay. The number of nucleus. You have the multinucleated muscle fibers in skeletal muscle. In cardiac muscle, we have the mono or binucleated. Then in smooth muscle, we have the mononucleated, muscle fibers. The mechanism of muscle contraction. For skeletal muscle is actin based. For cardiac muscle is actin based. For smooth muscle is myosin based. Why? Because now in skeletal and cardiac having the same mechanism of contraction, there is actually actin. Actin is a determining factor for contraction. Why in smooth muscle, myosin is a determining factor for contraction. Okay. Then finally, location. For skeletal muscle is associated with skeletal tissues like bones, like cartilage. In cardiac muscle is found in the heart. That's the location. Then the smooth muscle is found majorly in the visceral organs. Now let's have a short break now. I'll be back after this timeout. Alright, so welcome back. Let's talk about the fifth topic, muscle contraction. Muscle contraction simply means shortening of muscle length. Better presented, it is a alteration in either the length or tension of the muscle fibers. Okay, types of muscle contraction. The fascic contraction, which occur within a short period of time. Tonic contraction, which occur for a longer period of time. Okay, which is responsible for muscle tone. Muscle tone is a continuous partial muscle contraction, which tends to resist muscle stretch. Okay, the fascic contraction is further divided into isotonic contraction and isometric contraction. Isotonic contraction is in which case the muscle tension is constant, but the change in muscle length produces muscle movement. Okay, but there's a change in muscle length, so the muscle tension is constant, but there's change in muscle length. Okay, so this one produces movement. In isometric contraction, the muscle length remains the same, but there's a change in muscle tension. Okay, now let's talk about isotonic contraction. Isotonic contraction is further divided into concentric contraction and eccentric contraction. Concentric contraction is when the muscle fibers contract towards the center of the action. Okay, so when you flex your elbow joint, the biceps brachii muscle fibers, they are undergoing concentric contraction, while the triceps muscle are simultaneously undergoing eccentric contraction. Okay, so eccentric contraction is muscle fibers they disperse away from the center of action. So usually when an agonist muscle is contracting concentrically, antagonist muscle will be contracting eccentrically. Please note that. All right, let's talk about skeletal muscle contraction. Excitation contraction coupling. Excitation contraction coupling, it's simply the stimulation of skeletal muscle fiber by a neuron induce or generate contraction. When a skeletal muscle fiber is excited, then it contracts. Okay, so coupling of the two things together is what we mean by excitation contraction coupling. Now let's talk about the steps involved in the contraction. Now we have discharge of motor neuron. That's number one. Number two, release of neurotransmitter at the motor end plates. Number three, the binding of acetylcholine to nicotinic acetylcholine receptors. Number four, the increase in sodium and potassium conductance in the end plate membrane to generate the end plate potential. That is the form of gradient potential. The generation of action potential in muscle fibers. So there is generation of action potential in muscle fibers. Okay, that's the sixth point. Then some point is there's inward spread of depolarization along the T-tubules. Okay, then we have the release of calcium ions from the terminal systems of sarcoplasmic reticulum and diffusion to thick and thin filaments. Okay, then we have the binding of calcium ions to troponin C. to uncover the myosin binding sites of actin. Then there's formation of cross linkages between actin and myosin, and the sliding of thin on thick filaments to produce shortening. That's the movement that you see now physically. Now the steps in relaxation. We've just talked about the steps in contraction. Steps in relaxation. Calcium ions are pumped back into the sarcoplasmic reticulum. Then there's release of calcium ions from troponin. Then there's cessation of interaction between actin and myosin filaments. Okay, now that's for skeletal muscle contraction. Now for cardiac muscle contraction, it's simply the same mechanism because both are striated. Okay, the little difference is cardiac muscle depends on extracellular calcium ions for contraction. So why the skeletal muscle depends solely on the calcium ion in the sarcoplasmic reticulum, cardiac depends on extracellular calcium ions, okay, for its contraction. Then muscle contraction is longer in cardiac muscle than skeletal muscle because of slow calcium channels that causes plateau, in cardiac muscle fiber. So consequently, cardiac muscle tends to have long absolute refractory period, which prevents its tethanization, its fatigueness, and its summation. Okay, that we've just talked about when we're trying to differentiate between those muscles. Alright, the smooth muscle contraction. Now let's talk about the mechanism of smooth muscle contraction. Arrival of action potential in the smooth muscle fiber causes the opening of calcium channels. The calcium ions binds with calmodulin. So the calcium calmodulin complex joins with myosin kinase, a phosphorylating enzyme, and activates it. Okay, so the activated myosin kinase enzyme now phosphorylates myosin light chains and permit it to bind to actin. Okay, now actin and myosin subsequently interact by filament sliding to produce contraction in a similar way to that of of skeletal muscle that we described previously. Smooth muscle is able to maintain a high force of contraction for longer period of time with little ATP usage. That's through the large phenomenon, the ability of the smooth muscles, especially the smooth muscle in the GIT, maintain a high force of contraction over a long period of time with little energy usage. Okay, please note that the thin filaments are acting associated with tropomyosin, and so there is absence of troponin system in smooth muscle fibers. Please note that. Then the thick filaments composed of myosin only binds to actin if the light chain in the myosin is phosphorylated. So that's why myosin is a determining factor for contraction in smooth muscle. You only have the binding of myosin with actin only if the light chain is phosphorylated. Okay, now the cessation of contraction, that's relaxation. Now we talk about the role of myosin phosphatase. Why myosin kinase tends to phosphorylate the myosin light chain, myosin phosphatase tends to dephosphorylate the myosin light chain, so that reverse of the contraction is performed and there is induction of relaxation. So the myosin phosphatase tends to withdraw the phosphates from the light chain, and myosin return back to its initial state, so it no longer binds with actin. Alright, now let's go to the sixth topic, which is the membrane potential. Now the resting membrane potential is the membrane potential at the cell resting state. Now the normal value is minus 70 millivolts in neurons and minus 85 to minus 95 millivolts in muscle and some large neurons. Okay, now let's go to membrane action potential and membrane gradient potential. Membrane action potential. Action potential is an all or non-electrical event which occur along the membrane of an excitable cell as a result of influx of ions, okay, mainly sodium ions, into the cell. Alright, let's talk about the phases. We have majorly the depolarization and repolarization phase. Now, action potential curve. We have the latent period during which we have stimulus active fat, that is a short deflection, in the graph. We have the firing or threshold level, in which case when the gradient potential becomes action potential. A firing or threshold level is the level when the action potential begins. Depolarization, we have overshoots, we have repolarization, we have after depolarization or negative after potential, then we have after hyperpolarization, which is called positive after potential. Alright, now gradient potential is a localized non-propagate potential that causes a mild or little change in the membrane potential. Okay, that is a gradient potential. Now let's have some differences between action potential and gradient potential. Action potential is propagative, that it's spread all over the excitable cell, while gradient potential is non-propagative or is localized. Okay, now action potential has both depolarization and repolarization. Gradient potential has either depolarization or hyperpolarization, so it goes in one direction alone. Okay, action potential obeys all or none principle. Gradient potential does not obey the principle. Action potential does not allow summation. Gradient potential allows summation. Action potential has refractory period. Gradient potential does not have refractory period. Action potential cannot reverse back to gradient potential. It cannot reverse back. Gradient potential can give rise to action potential. Okay, gradient potential can give rise to action potential. Action potential does not respond to strength of stimulus. It only responds to the frequency of adequate stimulus. Okay, but gradient potential responds to strength of stimulus. Okay, now action potential is generated only when there is adequate stimulus or threshold stimulus. Okay, while gradient potential is generated even with subminimal or subthreshold stimulus. Okay, can also induce gradient potential. Now let's have examples of gradient potentials. We have end plate potential, we have electrotonic potential, we have receptor or generator potential, we have excitatory and inhibitory postsynaptic potentials. Okay, now let's go to the seventh topic, which is synaptic transmissions. Synapse is the communicating junction between two or more neurons. Okay, types, we have electrical and chemical synapse. Electrical synapse, it involves the presence of gap junction and it allows the flow of ions directly from cell to cell. It's located in the heart and GIT muscles. Okay, chemical synapse is mediated by chemical substances that we call neurotransmitters, which are released into the synaptic cleft from the axon terminal to the postsynaptic membrane. Now, talking about synaptic transmission, arrival of action potential at the axon terminal will generate the release of the neurotransmitter from the synaptic vesicles, okay, into the synaptic cleft. When the neurotransmitters are released into the synaptic cleft, they bind to their receptors on the postsynaptic membrane. For the synapse, before the synapse, you have the presynaptic neuron. After the synapse, you have the postsynaptic neuron. Okay, so now that's what synaptic transmission is all about. So it's just the transmission of impulse of signal across the synapse from the presynaptic neuron to the postsynaptic neuron via the use of neurotransmitters. So the neurotransmitter binds to its receptor on the postsynaptic membrane. Okay, now let's talk about the neurotransmitters. We have acetylcholine, we have some amino acids as we have gamma aminobutyric acid, that's GABA, we have glycine, we have glutamate, we have aspartate, those are amino acids. Okay, then we have some amines too that are neurotransmitter. We have adrenaline, dopamine, serotonin, histamine, those are amines. Okay, the first three are catecholamines. Mechanism of neurotransmitter inactivation in the synaptic cleft. We have some mechanisms in which the neurotransmitters are inactivated in the cleft. Number one, we have migration. Number two, we have phagocytosis. Number three, we have reuptake. Number four, we have diffusion. So you can use something like Mr. Pre-degree. So migration, phagocytosis, reuptake, and diffusion. Now, a motor unit is the combination of axon nerve fibers with all the muscle fibers that it innervates. Okay, that's a motor unit. Now, axon extends to different muscle fibers, okay, to innervate them. So the axon plus the muscle fibers, the number of muscle fibers that it innervates, is collectively called motor units. Okay, now let's go to the 8th topic, which is talking about the pathophysiologies. Now let's start with the myasthenia gravis. Myasthenia gravis is an autoimmune disorder. There is immunity against the acetylcholine nicotinic receptors in the postsynaptic membrane. Okay, that causes myasthenia gravis. That is loss of strength. Now, a similar condition is Eaton-Lambert syndrome. Eaton-Lambert syndrome, in which case there's also autoimmune disorder in which there's immunity against the calcium channels in the axon terminal. That is the presynaptic neuron. In the axon terminal. Okay. Now we have hypertonia, that is high muscle tone. We have hypotonia, that is low muscle tone. We have fatigue, that is decrease in muscular activity as a result of repeated stimulation. It becomes desensitized. Okay, that's fatigue. Then we have tetanus. Tetanus is a continuous muscle contraction. Okay, we can have complete and incomplete tetanus. Complete tetanus is there is no slight relaxation in between. Okay, just have contraction continuously like that, without even any slight relaxation. But incomplete tetanus is when you have little relaxation in between the strong muscle contractions. Okay, so that's actually end the whole section. I hope you enjoyed the presentation. Do get our other presentations on physiology. We succeed.